Just a quick reminder, guys, we were going to start a little bit later today, about 9:10, only because a couple of people had commitments till 9. So hang tight. I'm going to come back and we'll start as soon as I see those two people in particular sort of join us. Madison was one of them. Julia also was getting off work at 9. So I'll join in as soon as I see everybody who was planning on being here. So guys, I'll just wait until I see Julia join us. I know she had indicated she was going to be a little bit late, so I'll wait to get started. I'll just wait another 2 minutes here, guys. I know we had told Julia we would start at 9:10, so I don't want to start early without her. Well, guys, I'm going to go ahead and get started. I want to make sure to respect your time as well. Can I see you guys? Say hi. Hi, Madison. Are you with Maria? Hi, Lee. No, you guys are in different places right now. All right. Hi, Allie. All right. So, oh, look at the kitty. Oh, is Liv with you? No. Okay. I couldn't see whose hand that was. What's your kitty's
It was
name?
my roommates. Maple. This is Maple.
Daisy, come here.
This is my roommate, Katie.
Hi.
Hey, I'm Madison.
All right. Daisy. Daisy, did you bring a baby? Oh, you and Charlotte brought babies. Oh, I know that you're rough with your baby. Okay. All right, well, let's get going here so that I actually think I'll probably be done by 10 anyways, because this is one of the more condensed sort of learning units. So I'm going to share my screen here. And let's see. I will share this out. Is that Julia? I can't see here. Let's see. Yep, that's Julia. So I will share this document out as soon as we're done. It's again one of those pieces that sort of shows you the starting question, so to speak. And I'll highlight here that the bulk of the exam, like about half, a little more than half of the exam, is sort of this compare and contrast type things. So I'm going to be asking questions about electron transport, oxidative phosphorylation in mitochondria. And a question you might see is, where does electron transport occur in an animal cell, right? And so then we're asking specifically about electron transport and photophosphorylation in chloroplasts. So again, electron transport occurs in the inner mitochondrial membrane. What small molecule is the ultimate source of electrons for the electron transport chain? That's water, right? So those are the kinds of questions you're going to see. But really asking you guys to think about comparing and contrasting what we see with ETOP and mitochondria. That was learning unit 5. And actually, these numbers are wrong. It should be 5 and 6 here. And then electron transport and photophosphorylation in chloroplasts. So then there's some short answer questions, thinking about comparing and contrasting equations that we have for fuel oxidation versus equations that we have for the light and dark reactions. So again, I'll show you where those slides are to think about. And again, pathways for electrons that we have. Think about this as being sort of a compare and contrast. What things are similar, what things are different. Think about when we sort of role played that and we went through electron transport and oxidative phosphorylation in mitochondria, and then we transitioned to thinking about it in chloroplasts. And some of you had the exact same role, right? And some of you, like Maria, had no role in photosynthesis, right? Because complex 2 was not in chloroplasts. And again, a compare and contrast sort of structure and anatomy between mitochondria and chloroplasts. And then, like I introduced in our last section, you're going to see some new questions ask you to think outside of the box. This time there's going to be 3 that you pick. There's still a list of questions to pick from, but there's a few more that you have to sort of pick from. So pretty, fairly structured kind of content here. I'm going to come over and we'll just kind of go back and forth. We'll look at the study guide. I might tell you that there could be a few things that I've taken off the study guide, so I'll make sure to sort of take a look at that. And even at this, I haven't even split it up in terms of Unit 5 and Unit 6 as sort of like separate entities, because really we sort of learn these as very integrated, a lot of compare and contrast, and that's sort of what we have here. And again, thinking about the 2 main equations that describe both fuel oxidation and then carbon fixation. So maybe it's worthwhile. Let me come up here. I'm going to move this down just a little bit so I can access things. If we look at sort of the notes that we have here, the slides that we have for photosynthesis, again, this is one of those compare and contrast, fuel oxidation versus fuel production, right? Another compare and contrast has to do with mitochondria versus chloroplast. So you'll sort of see a lot of these compare and contrast pieces. Fundamentals, we didn't have any fundamentals for photosynthesis, but we had an electron transport oxidative phosphorylation sort of fundamentals quiz. So use that as a study tool as well. I haven't clearly gone through and fixed a lot of things here from using dark reactions. I really want to encourage you guys to think about as light dependent and light independent reactions, because I don't want us to think about, oh, that occurs at night. Again, sources of electrons, and then acetyl-CoA as a branch point metabolite. So that is really more, let me go over to the electron transport slides here. Thinking about where electrons come from, and then the importance of having acetyl-CoA as a branch point metabolite. So particularly in the beginning sections of slides that we're going to see with these future learning units, hopefully a lot of it is a review for you. So hopefully those couple of slides there were reviews. Again, there's not too much on this study guide here, so I'm going to pause periodically. I'll just see with these general equations, comparing and contrasting, any questions on things? Again, I'm going to go through the homework as well too and highlight where things are there. Remember, these numbers here highlight, if you're looking at homework questions, which homework questions to kind of take a look at to understand those concepts. Thinking about organelle structure, compare and contrast, mitochondria and chloroplast structure. Thinking about that endosymbiont theory, there's not really too much you need to, focus on with that. The concept was really just thinking about how we had both mitochondria and chloroplasts. I guess that was really more, we started with that theory in mitochondria, sort of highlighting that they were an ancient bacteria that we had there. So this is the endosymbiont theory, that we had early prokaryotes that got swallowed up by another prokaryote, and that generated eukaryotes. So being able to sort of think about those compare and contrast pieces there. Again, structural details, comparing and contrasting mitochondria and chloroplast. Thinking about some details about things that we need to get where, right? And so we talked about this. I'm going to highlight what slide that is. We talked about that slide really here. This is sort of the big slide that talks about how do we get things where they need to go, particularly if we don't have a door to get them there. So thinking about getting oxaloacetate across the mitochondrial membrane, right? That's really going to be sort of the malate aspartate shuttle that we had there. Thinking about the facilitated diffusion process for getting ADP and ATP to sort of exchange where they need to be, reminding ourselves that that also contributes to the charge gradient. And then thinking about one other way that we can utilize that proton gradient is we are able to piggyback in that inorganic phosphate that we need for ATP synthesis. Let's see. So that's sort of thinking about the structure of those organelles, chloroplasts and mitochondria, and thinking about some of the players that's involved. So I'll pause there for a second, see if anybody has any questions. So highlighting when we talk about comparing and contrasting electron transport as it occurs in chloroplasts versus mitochondria, I want you to think back to sort of the role-playing activity that we had when we went through and talked about how the electrons move through the electron transport chain in mitochondria, how that's different in chloroplast. So again, some semi-detailed questions about those pieces. Thinking about the energetics piece, right? We see most of the compare and contrast pieces when we talk about photosynthesis. I'm going to come back to this slide because I'm recognizing it's not a slide that we talked about. So I'm going to just come back to that and just highlight a couple of things. Not really anything detailed that we didn't see elsewhere, but we didn't specifically talk about it on that slide. So this is again a compare and contrast about the electron transport chains between photosynthesis or between chloroplasts and mitochondria. So we talked through these guys a lot when we talked about where do electrons come from, how are they high energy, where do we pump a proton gradient, things like that. So let's see, pathways of electrons. So when I talk about pathways of electrons, this is what I'm talking about. These sort of flow charts here, if you will. You know, where do electrons? So I want you to know sort of the pathway of electrons for both chloroplasts and mitochondria, thinking about sort of where they start, where they end up, so forth. And again, who is able to translocate protons? So we don't want to talk about, you know, we have Julia Wiggle. That is not the appropriate answer for how complex one translocates protons, right? I want you to talk about pKa shifting and so forth. Let's see. Light harvesting. This is the piece I'm going to go back and just sort of talk about very generally in our notes. We sort of talked about this a little bit, but I want to just make sure to have a picture to associate with it. And then being able to connect the specific products that we sort of generate through electron transport. So I'm going to come over just to our photosynthesis notes just for a second and highlight what we have here. We talked about this a little bit, just not with this slide. Okay. With both photosystem 2 and photosystem 1, we sort of talked about it's really the chlorophylls that are there. They are what grab onto that light energy, and then electrons get excited. So this is just an interesting piece that sort of highlights the different pigments that are found within plants. And specifically, it's chlorophyll A and chlorophyll B that really correspond to most of the green pigments that we see within photosynthetic organisms. This piece here, the only thing that we did talk about this, we said why we have P680 and P700 is we have to match the energy gap to a certain wavelength of light. So if you want to excite an electron from this chlorophyll up to its excited state, it's a defined energy gap, and that's going to be matched with a certain wavelength of light. So we talked about that. It's either P680 or P700. Now, the other piece that we really didn't talk about, and it's not really important for you to know it for the exam, so I'm not going to really go into details, but there's lots of chlorophyll pigments that are within plants that harvest light energy, but they are not actually the ones that eject the electrons. And so that's what we have here, where we have there's all of these antenna pigments, and then eventually you'll get to that magic chlorophyll that gets to be the one that gives up its electron. But not anything you need to worry about. There's not specific questions that are on that. But I did like this picture because it reminds us how we have to match energy gaps in order to absorb specific wavelengths of light with chlorophyll. So I'm going to come back to our study guide for a second. I'll pause for a second, see if anybody has any questions on sort of the general aspect, compare and contrast equations for chloroplast and mitochondria, organelle structure, compare and contrast, or electron transport, compare and contrast. Any questions on any of that? Okay, the last little piece here that's talking about pathway details, and then we'll sort of get into some regulation, integration, and application pieces. Um, Think about the steps of oxidative phosphorylation and then photophosphorylation in mitochondria and chloroplasts, right? So this was Olivia. She basically, had the same ATP synthase in both chloroplasts and mitochondria. So that is basically the same. So we didn't even talk about it. I see there's a question there. I'll get to it in just a second. Um, that is thinking about sort of this slide, right? And we only had to go through this slide once because it's the exact same machine that synthesizes ATP in chloroplasts. Let's see. Maria wants to know, can we go through the energetics of electron transport? Yes. Okay. So probably a great way to think about it, since it'll allow us to compare and contrast, is looking at this slide. Okay. So when you think about the energetics that we have here, so we're looking at the energy level of the electrons as a function of progress through the electron transport chain. So that's what these two little graphs are sort of showing. I want to highlight that we did sort of have, where was it? I'm trying to find this slide. Oh, here. This is the same kind of pictures here, but it's showing it sort of against this piece. So Maria, what I want you to imagine with this piece for electron transport, imagine I take these complexes and I don't stack them one on top of one another. Imagine I sort of lay them out like they happen in progression. And that's essentially what we have here. We start with the electrons at NADH that are high energy by virtue of their chemical composition. And as we go downhill, right, that's what we're showing here, is going downhill. Now, rather than showing like a progression over time, this diagram just stacks everything one on top of one another to show how you go down. Okay. But what I want you to remember is there's certain complexes that are capable of contributing to a proton gradient, right? So complex 1, complex 3, complex 4. But every time those electrons go through one of those complexes, they end up lower in energy. So that's sort of what we're thinking about by the energetics. So in mitochondrial electron transport, electrons start out high energy by virtue of their chemical composition. So NADH has a very large negative standard reduction potential. So it's high energy. And that's different from what we see when we talk about the energetics of electron transport in chloroplasts. And in that case, our electrons ultimately start out, not ultimate, like ultimately they start out coming from water. Water is a very low energy molecule. Now, we talked about, and we used Julie as an example because she was photosystem 2. She had a chlorophyll inside of her that got an excited electron because it was exposed to sunlight, and then she ejected that electron into the electron transport chain. Now, again, that electron is high energy by virtue of being exposed to sunlight. It excited it. Okay, so the big concept when we think about this is this energetics piece is, why are those electrons high energy? It's either because they're high energy because they're part of a chemical compound that makes them high in energy, or we use light energy to excite them to be high energy. Okay, does that help? So that's sort of the compare and contrast piece. Now, within electron transport in chloroplasts, we actually have a double bump. We excite those electrons twice so that they can end up at the end being in a higher energy state than they started. Notice again the compare and contrast. We start with a B vitamin derivative that goes down lower energy to water. Ultimately, in chloroplast electron transport, we start with a low energy water and we end up with a high energy B vitamin. Make sense? Does that help? Okay. Um, So that's the ATP and carb. Oh, I didn't finish that. So ATP synthase structure, we sort of talked about that. The binding change mechanism. So that was sort of getting back to, let me come here. It was on photos or the electron transport piece. So sort of knowing this piece, how that ATP synthesis machine works. Hang on, I have to wait for that menu bar. There we go, to go away. Comparing and contrasting pentose phosphate pathway versus Calvin cycle. You do not need to know details of this. It is really this slide at its core, right? So many, and this is why I do both of these learning units always back to back, always as part of the same exam, because there's so many things where one reaction, one process moves an equation, in one direction, and then the other process moves it exactly in the other direction. Now, I would not expect you to know the entirety of this equation, but I do want you to remember, purpose of pentose phosphate pathway. I use carbohydrates and I generate NADPH and carbon dioxide. The opposite happens in the Calvin cycle. I use carbon dioxide and I fix it together using the glue in NADPH to generate carbohydrates. So fundamentally, that's all that I really need you to know about comparing and contrasting those pieces. Rate determining steps. I don't actually don't we really didn't talk about that too much. I do want you for electron transport, I should say. For photosynthesis, we did sort of talk about the importance of three enzymes. We talked about rubisco, and then we talked about FBPase, these guys. These are the three enzymes. I want you to remember their names from photosynthesis, right? From those light independent reactions from the Calvin cycle. These are three enzymes that are rate controlling enzymes for the Calvin cycle. And we went through and we talked about how we control whether they're active or not. And these are new ways to sort of control whether or not something is active. For these guys, we previously had just learned about phosphorylation events and binding small molecules. These guys are turned on or off by whether or not cysteine residues are in an oxidized or reduced state. So I'm going to flip here just for a second and remind you, this was sort of this electron hot potato piece, right? If light's going on, there's a lot of electron transport going on. There's lots of electrons that can reduce these two enzymes. They are active in their reduced state, okay? And then we highlighted for this super important enzyme, rubisco. Rubisco is controlled by three things. It's almost like there's like a three checkpoint that if we're going to make sure that rubisco is active, we need to make sure that certain things are happening. The first thing we have to make sure is that we are doing a lot of electron transport, which means the pH in the stroma is going to be high. Remember, rubisco lives in the stroma. Okay, so if pH is high, that's going to be activated. So rubisco is an enzyme that's activated under high pH, low proton concentration. Remember, if we're pumping protons across from the stroma into the inner membrane compartment, okay, magnesium is going to flow in the opposite direction because magnesium can go across that membrane. It's going to want to go in the opposite way as that positive charge. This also tells us that magnesium is going to activate rubisco. So all of these signals that say, hey, electron transport is going on, you better be activating the enzymes, all three of these actually, that are responsible for doing the light independent reactions. And then the last piece here just has to do with whether or not a transition state analog is synthesized that could inhibit, right? Compare and contrast what we've talked about. We talked about Olivia. She was our ATP synthase machine. Let me go to those slides just to show you. We said, how do we make sure that she is not undoing her ATP synthesis work by dissipating a proton gradient and moving protons in the wrong direction if we're not doing electron transport? And again, there was another sort of way that we were inhibiting this, and that has to do with binding this inhibitor molecule. We call it IF1, and it's pH dependent. So just like rubisco is controlled by pH, ATP synthase is controlled by pH as well in mitochondria. So let's see. I think that sort of answered the questions that I wanted to get to. I keep hitting my menu and then I have to wait for it to go away. Carbohydrate synthesis routes. We didn't go over this. So I'm just going to highlight the things that I would sort of expect you to know because they're themes that we've seen. Okay. Those are just some summary slides. This is a theme that we see. If you want to couple things together, okay, we don't just have like atoms say, hey, let's hang out and put some glue between us. We often have to activate something so that we have a good leaving group. Okay. We see that when we did glycogen synthesis. We see that here with plant starch synthesis. Okay. And so we see that we utilize ATP here as a way to add an OAMP group. So that OAMP group ends up being an ADP glucose molecule. That's a good leaving group. So we're going to have a good leaving group so that we can sort of couple these together. That is a theme that we've seen. So even though this is a new context, I still would expect that you could look at this and say, oh, yeah, that makes sense. I also want you to always remember we're going to see this as a theme frequently when we generate when we generate pyrophosphate. We've got pyrophosphatase. That is what's going to allow us to impart irreversibility. So some of these themes that we keep seeing over and over again, we're just sort of part of this. So those are some of the only things that I need you to know. I would still expect you to know sort of the structure of sucrose in terms of its glucose plus fructose. Cellulose has that beta-1,4 glycosidic linkage, okay? Here's the only other pieces that I want you to know. We've talked about this already, but just reminding you that here's the home here. Cellulose is a structural polymer in plants. Alpha amylose is a storage polymer in plants. And then sucrose is actually a transported carbohydrate in plants. So those are the three carbohydrates we tend to see in plants. And I want you to think and, contrast them to what we might see, for example, in an animal cell. So we had alpha amylose in plants, as that's plant starch. Animal starch is glycogen. Okay, so be able to sort of see those comparisons. We see cellulose with beta-1,4 glycosidic linkages. We see alpha-1,4 glycosidic linkages in things that we store in our body. And again, sucrose is, our table sugar. Let's see. So that's actually all that there was on this part to sort of discuss carbohydrate synthesis. So I'm going to pause for a second, see if you guys have any questions on sort of these pathway pieces before we sort of talk about some integrated regulation application pieces.
Yeah, could we go over the logic check in terms of pH and the concentration of H plus and how it affects our
Oh, for
cholesterol?
electron
That makes
transports?
sense. Yeah.
This guy.
Normally
Okay.
I get it, but it just I go the reverse way for this for some reason.
Super easy. So, Julia, just to highlight, sometimes one of the things that gets confusing is whether we're talking about pH or protons, which are the opposite. High proton concentration is low pH. So, again, and maybe it's one of those things that just you want to think about this IF1. Just remember that that's the inhibitor. Okay, so you want to think about the logic check. If IF1 is active, that means I'm inhibiting ATP synthase. Now, why you'd want to be inhibiting ATP synthase, that's Olivia and her lollipop head and all of that, is that will always move protons in a direction if allowed. Now, unfortunately, if it's moving protons back to the matrix, it needs to burn up, it's going to burn up ATP to do that, and we don't want to do that. So that's sort of why we need to turn it off. So here's what we have. When we have IF1 with 4 of them hanging out, it's inactive as a tetramer, okay? And if we start having high concentrations of protons, we're going to activate that inhibitor um, under those low pH conditions. So let's think about the logic check is, all right, that inhibitor is going to be active when there's lots of protons around. When would there be lots of protons around? Well, there's lots of protons around in the matrix when I'm not doing electron transport. Okay, if I'm not doing electron transport and I'm not kind of generating proton gradient, I probably have plenty of ATP around. What can happen in that case is Olivia will run in reverse. ATP synthase will run in reverse and it will actually burn up ATP. And we just don't want that to happen. That would create a futile cycle. Does that help? All right. Anything else on these pathway details before we sort of go on to the next piece? All right. So, couple of the things I am going to tell you right now that I'm taking off because we didn't talk about it. You can cross this piece off. We did not talk about an ox-phos ratio, okay? But we did talk about uncoupling proteins. So part of this question I'm just going to highlight. We did not talk about an ox-phos ratio, but we did talk about uncoupling proteins. So we've talked about multiple ways that we can disconnect a proton gradient from synthesizing ATP. We talked about uncoupling proteins, okay? And we talked about diet pill, 2,4-dinitrophenol. And we actually just a little extent sort of talked about the piggybacking sort of process. That's how we get inorganic phosphate where we need it to go. And then Julia just asked the question, so we actually just addressed the whole regulation by the IF1 inhibitor. Some of these sort of big piece compare and contrast, not between mitochondria and chloroplasts, but between chloroplasts and mitochondria that exist in plant cells. This is one of my favorite slides. I love this story, thinking about the difference between night and day for plants. And those of you who were at Dr. DeLance's talk, I loved that he sort of talked about that, that is a pretty cool thing that they actually behave like animals at night, and that's why they need oxygen, right? It's a fallacy that plants don't need oxygen because when the sun is not out, they need to behave like an animal. So this is sort of an important big picture slide here. We did already talk about this, but I'll highlight again the regulation of Rubisco. Let me sort of come back to where we had that. So again, here's how we control Rubisco. I have it on this slide sort of written down here, but here's the words of it too. So if you were to think about Rubisco and you were going to ask, what makes it most active? What makes it most active is a high pH, high magnesium concentration, and the absence of that transition state analog, right? So think about those kinds of questions where you could think about, hey, this is like a 3-part system to sort of control the activity of this enzyme. So that's Rubisco. We just talked about the FPBase, SPBase regulation, and then just reminding ourselves that obviously we don't want glycolysis to be going on as a mechanism to generate ATP during the day. So let me go back to that slide here for a second. We do not want, actually it's probably part, where is that slide? Here. We obviously, this is glycolysis that's going on, right? We do not want glycolysis to be going on and breaking down carbohydrates to ATP at the same time that the Calvin cycle is happening and taking ATP and NADPH from the pentose phosphate pathway and making these. What we've got shown in red here would be a futile cycle. Definitely don't want that. And then this concept of time versus space. This was sort of like this last piece that we talked about. I do want you to remember that plants need to use oxygen when they're using their mitochondria and doing that kind of chemistry. That's usually at night. They can use oxygen during the day in this process called photorespiration. Respiration meaning using oxygen, and they're doing it in their chloroplasts. And why they do it is Rubisco is so hyperactive because there's so much electron transport going on that it needs to do something. And so even though it's inefficient and is kind of a futile cycle, if you use oxygen as a substrate for Rubisco, you at least allow those electrons to sort of keep flowing, because if you back up those electrons, that can cause oxidative damage, and that can be irreversible in a plant. So another way that oxygen can get used is when it's light out in this process of photorespiration. So then the other concept is if plants need to pull in CO2 to do carbon fixation, and that happens when it's light out, well, often when it's light out, it's also really hot out. And the way that they let CO2 in is through their stomata, through their pores. It allows for gas exchange. Unfortunately, that can allow water out as well. So what we can do is we can say, hey, let's harvest carbon dioxide at a time where it's better to bring it in, like maybe at night, but we need to stockpile it. And so it's a way to sort of create this hidden reserve of carbon dioxide by putting it together with phosphenylpyruvate and making the 4-carbon molecule oxaloacetate. We reduce that to malate, and malate is what's stored. Okay. This is why this process is called a C4 plant. Will do this is because it's making and storing carbon dioxide in a 4-carbon molecule. C3 plants, and I think I have another slide on this, just sort of summarize it. C3 plants immediately use that carbon dioxide and fix it as 3-carbon GAPs. These C4 plants are going to fix it and store it and for later use as the 4-carbon malate. So I want you to know the difference between a C3 and a C4 plant. Okay, that's again different from this photorespiration piece, but still tends to be things that happen with plants that are exposed to a lot of sunlight. Okay, but the reason they're doing this is they need to find a way to stockpile and store carbon dioxide at a time when it's not going to be problematic. Okay, they do this by having 2 different cell types. Okay, so they sort of compartmentalize and sort of store that in those mesophyll cells. And then when they're able to, and they can store that at night. And then when it's like light out and they're doing a lot of electron transport and they want to be doing that Calvin cycle, they can actually generate that carbon dioxide here. So here we see they generate that carbon dioxide here, and then it goes into the Calvin cycle. Okay. So I want you to know the difference between C3 and C4 plants, why we sort of separate things in space. And then, so you can have C4 plants that still are going to collect carbon dioxide, but they might do it during the day, right? They just store it in different cells. And then you can also have those plants that live where it's really, really dry, and they'll actually separate it not only in space, but in time as well. And that's sort of what this is meaning over here, this time versus space sort of situation. I already mentioned this concept of how and why photorespiration occurs, and then we just sort of talked about C3 and C4 plants, and then CAM plants are a special kind of C4 plant that also separates not only in space, but in time. So I'm going to pause there for a second because I went through a lot that was there, and see if anybody has questions on any of that stuff. Again, I'm still going to flip through slides and homework and see if there's anything that I want to draw attention to that I haven't mentioned yet. Some of these things are repeats of things we've already talked about when we think about the phosphorylation cascade versus redox cascades, right? That is the slide that's here. Again, you really don't need to remember this for right now, but it was reminding you that, hey, we've talked about phosphorylation cascades as a way to regulate enzyme activity. This is just a redox cascade. So we're not having multiple kinases add or remove phosphates. We're having kind of the hot potato of electrons going through. This is something that I didn't specifically talk about. I don't think I saw a the cyclic versus non-cyclic. So this is worth kind of reminding ourselves again that ferredoxin has 3 options for what it can do with electrons. We have a non-cyclic pathway where it just continues those electrons onto its ultimate final electron acceptor as NADP+ to make NADPH. Or it can go and deliver them back to Connor. Connor was our Q, so it can deliver them back there. Cyclic pathway allows us to generate a bigger proton gradient, make more ATP. And then we highlighted that especially when it's very light out, you want some of these electrons going through this redox cascade so that you can ensure that the enzymes FPBase and SPBase are able to do their chemistry in the Calvin cycle. Let's see. You can skip over this because we really did not talk about that at all. And let's see, we actually didn't talk about any of these at all. So this whole last section, don't even worry about it. We didn't get to it. Not important. Any quick questions before I start just kind of making sure that there isn't anything else I want to discuss either from homework or notes?
Did we go over what a CAM plant is in class?
So I may have mentioned it briefly, so let me just go over just a little bit more detail. So Connor, I'm just going to ask you questions since you asked about it. The difference between C3 and C4 plants, does it make sense? So again, just to highlight, C3 plants immediately fix carbon dioxide as the 3-carbon sugar GAP. And then we know we can make glucose and other things from it. C4 plants say, you know what? I need to stockpile carbon dioxide. So what I'm going to do is I'm going to fix carbon dioxide as a 4-carbon molecule called malate. And then when I need it, I can get that carbon dioxide back out. Okay. Now, CAM plants are a specific kind of C4 plant that not only do they have the separation in space because they say, I'm going to stockpile in a different cell type, malate as the 4-carbon piece, but it only collects the carbon dioxide at night because it's so dry out that it has to make sure that it separates at night. So CAM plants are a specific kind of C4 plant. Does that help?
Yeah, thanks.
Any other questions on anything we've gone over already before I take a quick glance through of our homework and then just go through our notes real quick, make sure there's nothing I wanted to highlight. All right, let's go through electron transport first. Again, encourage you take a look at your fundamentals quiz. Like those questions are certainly fair game in terms of the kinds of things you had to know for that. So that first part of your homework was the fundamentals quiz here. Especially as we see it, and I think there's a question in your photosynthesis homework, being able to think of the anatomy, compare and contrast between mitochondria and chloroplasts. So be able to think about that. We sort of skipped over because it was a lot of work to write this table, and I don't think you needed to have this table for the players that are part of the electron transport process. I really think we did a good job sort of going through it and thinking about it, just looking at this. And important sort of big picture skills are thinking about molecules that need to transit lipid bilayers, but we don't have transporters for them. So how do you do that? So oxaloacetate is one of those. And then again, we think about how we transport proton or inorganic phosphate, and then how we get ATP and ADP exchange. And that's sort of getting at those 4 transporters. Don't worry about this equation. We didn't go through any math. It was kind of on your slide deck, but so part of the idea here is to recognize that there's way more energy that's possible than we actually get out. So, but don't worry about that. You're not going to see any sort of electrochemistry type equations that we have here. So you can sort of ignore this question. This is just a pathway walkthrough, just sort of summarizing it. You don't need to know this equation, but I do want you to understand the concept that there's 2 things that can contribute to the energetics, the thermodynamics of electron transport. One is a concentration piece, one is a charge piece. For mitochondria, you get both. Okay. Now, what's the problem with chloroplasts? Because you're missing the ability to build up a charge gradient, your concentration gradient needs to be bigger in order to have the same effect, right? And we'll see that. I think there's a question on that on the chloroplast homework. Again, just reminding us of how ATP synthase works. Thinking about, now again, you do not need to know the OP ratio. We didn't talk about it. Fundamentally, all it means is the amount of oxygen used is related to the amount of ATP produced, because electron transport, final electron acceptor is oxygen, is connected to ATP synthesis. That's all that really means, but don't worry about it. You're not going to see any questions on that. We did talk about two molecules that we know can uncouple that process. Diet pill, 2,4-dinitrophenol, and then the naturally occurring uncoupling proteins. Julia asked that great question about thinking about the logic check for how we manage to inhibit ATP synthase when we're in sort of a quiet state. Cross off question 12, we didn't talk about obligate aerobes or reactive oxygen species. So questions 12 and 13, you don't need to worry about. Super fun stuff, but we just didn't get to it. All right, let me just flip real quick through our slide deck here, see if there's anything that I didn't cover. And if there's something you want me to sort of talk a little bit more about, we can do so. I think we've talked about all of these slides. We talked, and I know we addressed this at some point. We sort of talked about sort of conformational changes that happen because of protonation events. Yeah, and that was it actually for electron transport oxidative phosphorylation. Thoughts on anything that anybody wants me to go over? It's funny, as I was looking back over things earlier today, I said, you know what? I actually think it's probably about a 50-50 split on the exam, but I certainly know I spent more time sort of getting excited and talking about photosynthesis than I did electron transport and oxidative phosphorylation. All right, let me look at our homework questions for photosynthesis. Again, seeing the compare and contrast between fuel oxidation and then the combination of light and light dependent and light independent reactions to sort of do the opposite chemistry. That's those first three homework questions there. This again is that compare and contrast between what we see for the structure of mitochondria and chloroplasts. This was that question that I told you I want you to know sort of the pathway for electrons, right? And then Maria had asked about here, we can sort of see thinking about the energetics of how that works. This is just the pathway walkthrough, which we sort of did that live in class. Here, this question reminds us that we are missing sort of this ability to build a charge gradient in chloroplasts. And so you actually have to have a higher proton gradient in order to synthesize the same amount of ATP. And that's because of that permeability, specifically to magnesium and chloride ions. Let's see. Just reminding us that when we do cyclic versus non-cyclic pathways, it has to do with where ferredoxin takes its electrons. Cyclic allows you to deliver those electrons back to Q, and you can do more Q cycle, keep building up your proton gradient, versus non-cyclic, you favor NADPH production. And sometimes, depending on the needs of the cell, whether you need more ATP or more reductive biosynthesis currency, you can do either one of those. I really wouldn't worry about how these two questions are sort of worded here. I told you the details of what you need to know for comparing and contrasting it's right here, the pentose phosphate pathway and the Calvin cycle. This is sort of the level that you need to know. And again, it's related to what we see here. We produce NADPH and carbon dioxide. This is the sort of the piece that you need to know. Okay. And then in Calvin cycle, we're basically using carbon dioxide and using NADPH, but don't worry about sort of like the restructuring recycling piece, not relevant to what we need to talk about here. And then this recovery piece isn't important. Don't worry about question 12. You know, we really didn't talk about once you get to GAP from the Calvin cycle, how you get to other things. So don't worry about that. We just talked about it generically as carbohydrates. And then I do want you to know the 3 kinds of carbohydrates that are produced in plants. So we've got essentially a storage polymer, a transport sugar, and then a structural carbohydrate. So that's all that I really need you to know about those pieces. This is sort of the night versus day sort of piece, how plants obtain their energy. So question 14 is kind of an important question, particularly as it relates to sort of this picture, right? Day versus night for plants. So that's kind of one of those big picture sort of questions. I do want you to know the 3 rate controlling enzymes for the Calvin cycle. And then again, here is sort of the net of what happens in the Calvin cycle. We take carbon dioxide, we take some money, we take some glue, and we make carbohydrates. That's sort of the net of it. We went through this already, but sort of highlighting in detail how pH and magnesium levels can sort of control the activity of rubisco. And then there's obviously this inhibitor that's only synthesized when it's dark out. I'm not going to ask you about, you know, the glycogen-related phosphorylation cascade, but I do want you to be able to think about that phosphorylation cascade that we have for SPBase and FPBase. I do want you to remember those, and remember when they are in the reduced form, which means that cysteine is in its thiol form, they're going to be active. Then again, I just went through this just a second ago, talking about the different fates for where ferredoxin can take its electrons. Let's see here. This logic check of what's happening when it's light out or versus it's dark out, importantly as it connects to whether or not we want glycolysis to be happening there. We sort of went through that slide. We talked about photorespiration, again, another purpose for the use of oxygen. So again, if I were to ask a question and say, tell me 2 reasons why plants need oxygen. Okay. Obviously, they need oxygen because they do normal cellular respiration in their mitochondria. But the second piece is if it is very sunny out and they have depleted their local CO2 levels, rubisco will use oxygen as a substrate, and it allows us to continue electron transport so we don't have photooxidative damage. Went through this with Connor's great question, right? The differences between C3 and C4 plants, and then CAM plants being a very specific type of C4 plant. So that's what these last sort of 3 questions are here. Okay, and so CAM plants, not only do they regulate in space, but they also regulate in time as well. And actually, I think this is a subtlety that's not really important for you guys to know. I think the reason I crossed off in space, because they only harvest CO2 at night, they actually don't need to compartmentalize it someplace else. So they store it as malate, but because they separate it in time, they put it all together in one spot. Let me see if there's any photosynthesis slides, and then I think we will be good to go unless you guys have questions. Again, important slides here. Compare and contrast fuel breakdown versus fuel production. Mitochondria structure versus chloroplast structure. Comparative electron transport, both from an energetics and sort of a pathway standpoint. We went through that. We sort of talked about cyclic versus non-cyclic, the importance of having a greater pH gradient in chloroplasts. Again, these are opposite reactions, pentose phosphate pathway, Calvin cycle. And again, we clearly did not get into any of those details structurally. Day versus night, that's going to be an important sort of concept. How we control the Calvin cycle. Okay, rubisco, FPBase, SPBase. Know how those enzymes are controlled. Yeah, C3 versus C4. So that's kind of it. I know there's still a lot. I'm not going to say that there's not a lot that's there, but I think it's much more compartmentalized than some of our other exams. So I think students in general tend to find this exam a little bit more streamlined because there's certainly detailed content, but it's really in a very focused space. So questions?
For the charge gradient, are we going
Yes.
to know the two different values, like for mitochondria and chloroplast? No. Okay.
Nope. Nope. Just know that it must be significantly greater in chloroplasts. Anything else? Well, I will stick around for just a second here if anybody, has any specific questions. But otherwise, I will see many of you research students tomorrow. And then I will see the rest of you Wednesday for class, and then sometime on Wednesday or Thursday to take the exam. Okay? All right. We'll see you guys. Good night.
Thank you.
You're welcome.
I have two questions, not about the exam. Well, kind
You
of.
want me to let me stop sharing and let me stop recording.